Welcome to Semester 1 Cell Biology. Today, we are going to explore one of the most fascinating and dynamic structures inside living cells, microfilaments. If you have ever wondered how a cell can physically move, change its shape, or even divide into two, you are in the right place. We will build our understanding from the ground up, starting with what these structures are and eventually uncovering the elegant mathematics that control their behavior. Let us dive in. To understand why microfilaments are so important, imagine a large camping tent. Without the internal poles and tension ropes, that tent is just a floppy, shapeless piece of fabric on the ground. A biological cell is very similar. Without an internal support system, it would just be a squishy, fragile bag of fluid, unable to hold any structure. Microfilaments act exactly like the dynamic, flexible ropes of this cellular tent. But unlike a normal tent, these ropes are constantly being assembled and disassembled. This incredible flexibility allows the cell to change its shape, crawl across surfaces, and grab onto things in its environment. Now that we understand their purpose, let us look at what these ropes are actually made of. Microfilaments, which are also known as actin filaments, are the thinnest structural fibers of the eukaryotic cytoskeleton. They measure only about 7 nanometers in diameter. They are built from a globular protein called actin. When these proteins are floating freely in the cellular fluid, we call them G-actin. When they link together, they form a twisted, double-stranded polymer chain known as F-actin. A crucial feature of these chains is their structural polarity. This means the two ends behave differently. They have a plus end that grows very quickly and a minus end that grows slowly or even loses pieces. Knowing how they are structured helps us appreciate just how vital they are to human life. Without microfilaments, life as we know it would be completely impossible. They are the fundamental driving force behind muscle contraction. By working alongside a motor protein called myosin, they allow you to walk, breathe, and pump blood. Beyond your muscles, they are what allow your white blood cells to physically crawl toward infections to destroy invading bacteria. Furthermore, during cell division, they form what is called a cleavage furrow. This acts like a tightening microscopic belt that pinches one dividing cell into two new daughter cells. They are truly the microscopic muscles of the cell. Since these filaments are constantly growing and shrinking, we need a way to understand this balance mathematically. This brings us to the concept of critical concentration. Conceptually, there is a specific point where the filament is perfectly stable. The rate of new pieces attaching perfectly balances the rate of pieces falling off. We define this exact concentration of free actin monomers as the critical concentration, denoted by C sub C. In this formula, K off represents the rate at which monomers leave the filament, and K on represents the rate at which they attach. If the off rate increases, the critical concentration goes up. If the on rate increases, the critical concentration goes down. This delicate ratio dictates the baseline stability of the filament. Before we move on, let us see this formula in action with a concrete example. Imagine a researcher is studying actin filaments in a controlled test tube environment. She wants to find the baseline stability of the plus end. We are given that the rate constant for attachment, K on, is 12, and the rate constant for falling off, K off, is 1.4. We need to find the critical concentration. We choose our formula because it directly relates these two rate constants to the equilibrium point. By plugging in 1.4 for the numerator and 12 for the denominator, we set up a simple division. The result is 0.116 micromolar. Physically, this means if the test tube has exactly this concentration of free actin, the filament's plus end will remain perfectly stable, experiencing no net growth or shrinkage. Now that we have used this formula successfully, let us understand exactly where it comes from. The critical concentration formula is not just a random fraction. It is derived from the fundamental kinetics of actin polymerization. We start with the velocity equation, which states that the net growth rate, V, equals the rate of pieces attaching minus the rate of pieces falling off. To find the point of perfect stability, we set the growth velocity to 0. This is the mathematical definition of dynamic equilibrium. By substituting 0 for V and replacing the general concentration with our critical concentration, we can simply use algebra to rearrange the terms. We move the loss rate to the other side and divide by the attachment rate. This proves exactly why the critical concentration is simply the ratio of the off rate to the on rate. So we have proven how to find the stable point. This brings us to an important question. What happens when the cell is not at this exact stable point? To determine if a filament will grow or shrink, we must compare the actual cellular concentration of free actin, denoted by C in brackets, to the critical concentration we just learned about. We have two simple mathematical conditions. First, if the actual concentration is strictly greater than the critical concentration, the rate of addition outpaces the rate of loss, and the filament will grow. Conversely, if the actual concentration drops below the critical threshold, there is not enough free actin to maintain the structure. The rate of loss takes over, and the filament will shrink. These two simple inequalities govern the entire dynamic behavior of the cytoskeleton. Let us apply these growth conditions to a real biological scenario inside a human cell. We are given that the cell carefully maintains a free actin concentration of 0.5 micromolar. We are also given that the plus end of a specific filament has a critical concentration of 0.12 micromolar. We need to determine if this end will grow or shrink. We choose our inequality rules to compare the actual concentration to the critical threshold. Since 0.5 is significantly greater than 0.12, we see that there is plenty of free actin available relative to what this end requires to remain stable. Therefore, new monomers will rapidly attach. Physically, this means the plus end of the filament will experience continuous, rapid growth. Now that we have evaluated the plus end, let us look at the minus end of that exact same filament. The cellular concentration remains at 0.5 micromolar. However, the minus end has a much higher critical concentration requirement of 0.60 micromolar. Again, we compare the actual concentration to the critical threshold. This time, 0.5 is strictly less than 0.60. Because the available concentration is lower than what this specific end requires to maintain its length, there is simply not enough free actin to satisfy it. Monomers will fall off faster than they can attach. Physically, this means that while the plus end is growing, the minus end of the filament will continuously shrink and lose pieces. These two examples we just solved reveal a fascinating biological phenomenon. Because the plus end is growing, while the minus end is shrinking at the exact same time, the filament undergoes a process called treadmilling. Imagine a literal treadmill. The belt moves forward continuously, but the machine itself does not get any longer. The actin filament behaves the exact same way. It appears to move forward through the cellular space, even though its overall length might stay exactly the same. This is not just a neat math trick. This is exactly how cells push their membranes forward to crawl across surfaces. The cell is literally building the road ahead of it at the plus end, while tearing up the road behind it at the minus end. To truly grasp this concept, let us visualize the treadmilling process we just calculated. In this diagram, you are looking at a double-stranded actin filament in the center. Notice the distinct structural polarity we discussed earlier. On the right side, you see the plus end. Here, green G-actin monomers are actively attaching to the filament, represented by the green forward arrow. This is the growth phase, where the cellular concentration exceeds the critical concentration. On the left side, you see the minus end. Here, red monomers are detaching and falling away, represented by the red arrows, because the local concentration is too low to maintain stability. The large arrow at the bottom summarizes the physical interpretation. As pieces are added to the front and lost from the back, the entire structure effectively moves forward through space. We have covered a lot of ground today, so let us reinforce the most important ideas from this topic. First, remember that microfilaments are dynamic, flexible ropes made of actin that are absolutely essential for cell movement, shape changes, and muscle contraction. Second, the mathematical key to their behavior is the critical concentration, which is the exact balance point between monomer addition and loss. Third, by comparing the actual cellular concentration to this critical threshold, we can predict whether a filament will grow or shrink. Finally, this delicate mathematical balance allows for treadmilling, the incredible mechanism that lets cells physically crawl through their environment. You now have a solid understanding of both the biology and the mathematics of microfilaments. Great job!